right, thank you, David, for that comprehensive overview of ODX-ET. So in this portion of the talk, I'm going to go over some of the studies the team executed to not only demonstrate robust performance, but also prove it's safe and effective. So in this slide here, it shows a snapshot of some of the studies that we conducted. We had to extensively validate this assay, both analytically and clinically. And so you can see here are some of the studies, everything from establishing sensitivity and specificity to ensuring we have really good concordance to an already established orthogonal assay. And as you can see, some of these also centered around the specimen itself. So clinical FFPE samples are inherently variable, right? And so we did a lot of testing looking at tumor content and understanding the interfering substances that come along with it, et cetera. We also looked at a lot of different stability testing, so not only on the sample itself, but the reagent, ensuring it can be transported well, and also in use within the workflow. So some of these studies I'll go over in further detail, but let's dive into the first set of studies. So first we had to establish our limit of blank, essentially LOB, or specificity. And so this was essentially done to estimate the false positive rate for the assay across replicate testing in well-type samples. So in this study, we used 24 different well-type samples from clinical FFPE samples that spanned across 10 different tissue types, such as breast, colon, lung, etc. And these samples were thoroughly pre-screened using an established NGS orthogonal assay, because we really wanted to make sure these samples served as true blank samples for the assay. So the study was then conducted with 2 reagent lots and 4 replicates per tissue per lot. So a lot of replicates built into the testing. False positive rates were essentially calculated 2 ways, per position and per sample. So per position essentially just looks at the total sequenced positions. And then sample is just taking the total number of samples and replicates and using that essentially as a denominator for the calculation. So the false positive rate for the assay came out to be 0% across these 24 blank samples across these 10 different tissue types. However, we did want to note that there are some level 3 SNVs, false positives. The mean allelic frequencies for these SNVs were around 3.5%, so this was below our orthogonal assay's LOD and towards the lower end of our LOD. So in order to mitigate some of the false positive reporting issues, we went ahead and masked these level 3 SNVs from reporting. We then looked at limit of detection, or LOD, and essentially this was conducted by creating DNA and RNA variant blends across clinical FFPE samples, again with 10 different cancer types. We blended them to 6 different blend levels, so different variant allele frequencies, and the study was conducted using 2 reagent lots with 10 replicates. So this gave us over 120 data points per variant. LOD was conducted for both our CDX variant of interest, so EGFR exon 20 insertions, as well as a representative approach for our tumor profiling variants covering all of the main variant classes, SNVs, indels, fusions, etc. And so the detection rates across these different blends were analyzed using a probit regression analysis, and LOD was estimated. And so essentially what's the lowest allelic frequency, the assay can reliably detect 95% of that time. And so if you zoom into this portion of the table, you can see the LOD ranges that were achieved. And so we've got it broken down by our CDX variant, so EGFR exon 20 insertions, as well as our tumor variant classes, SNVs, insertions, deletions, CMVs, RNA fusion variants, as well as our imbalanced targets. And so just highlighting the CDX LOD ranges, we were able to achieve allelic frequencies of 3.24% to 4.38%. So both these studies together demonstrate really strong performance of the assay at very low detection levels. Next, we looked at precision, and we did this 2 ways. So looking at it both from a sample reproducibility aspect as well as panel reproducibility. For sample, that essentially just starts at the extraction level. But for the purpose of this talk, I'm going to highlight the assay the assay reproducibility. Okay, and then so we did this both for our CDX variants of interest, EGFR exon 20 insertions, as well as tumor profiling. So here, let's talk with the CDX variants. So repeatability and reproducibility was assessed using 4 EGFR exon 20 insertion variants. So these covered 4 different insertion lengths, 3, 6, 9, and 12 base pairs, and including wild-type samples. And we introduced variations that were sourced from different sites, different operators, and different instruments. And this generated over 144 data points per variant at 2 blended LOD levels. So near LOD, 1 to 1.5x and 2 to 3x. So different call rates were calculated, so no calls, positive and negative call rates. And so as you can see here, the positive call rate was 100% for all of our EGFR exon 20 insertion variants. And then also for our wild-type samples, the negative call rate was 100%. Repeatability was also assessed. For wild-type samples within repeatability was also 100%. And then for tumor profiling variants, right, we also assessed variability, right, across 3 external sites, 2 operators per site, and then 4 instruments per site. And so this gave us a lot of variation to look at. And so this table right here shows the positive call rates. And as you can see here on the left, breaks down by variant classes, or SNVs, insertions, etc. And then the 2 blended levels again, 1 to 1.5x and 2 to 3x. And so just zooming in one of the example classes for insertions, you can see that we achieved over 99.8% reproducibility for our tumor profiling variants, especially near LOD. In this next slide, it shows the negative call rate. And so again, just zooming in one of the variant classes for insertions, at near LOD, we got a negative call rate of 99.34%. Repeatability was also assessed for our tumor profiling variants, and a variance component analysis was performed, and the results showed a percent CV, or coefficient of variation, of less than 10% between operators and instruments. However, there were some exceptions to note here, 3 variants that came in a little higher than 10%. But overall, still really good repeatability, and then maybe not too surprising, our highest CV came from our RNA variants. Another notable study to highlight is our analytical accuracy. And again, this was done for both our CDX and our tumor profiling variants. And so essentially the accuracy of ODX-CT, or Oncomine Express Test, for detecting our EGFR exon 24 CDX variants or our tumor profiling variants was evaluated by comparing these results to an established orthogonal assay. And so for our CDX variants, this again was using an NGS-based orthogonal method. And then PPA, MPA, and OPA were calculated. So essentially, PPA was just looking at the agreement of ODX-ET positive calls with our orthogonal assay. And so here on the table to the right, you can see the results for PPA, MPA, and OPA. And so when compared to our orthogonal assay, the accuracy of EGFR 20 insertion detection was over 95%. And so you can see here for PPA 100%, MPA 95.5, and then overall 97.4. We then assessed our accuracy of our tumor profiling variants using a representative approach. And so this was actually conducted over 3 separate studies. So study 1 here is the bolus of testing. Over 460 or so FFPE clinical samples were tested in 15 or so different tissue types. And so as the table up here summarizes the results from study 1, and here to the left breaks down the variant classes. And then you can see over 586 variants were evaluated. And so it breaks it down by concordance of the ET positive calls to the orthogonal positive, and then the negative calls. And so even though there were some discordance with the orthogonal assay, we still achieved an accuracy of over 98.2% for all variants. And if you break down specific variant classes insertions, so it's like 100%. In study 2, this looked at additional level 2 variants, and specifically it was looking at ERBB2 CMVs in breast cancer, ALK fusions in lung cancer, and then RET fusions in both thyroid and lung. And this table down here summarizes the results from study 2. And so if we zoom in just here on the level 2 ERBB2 CNVs, we had really good concordance with the orthogonal assay, 29 positive and the 37 negatives as expected. We did have 4 discordance, but this resulted in 100% for the PPA and 90.2 for the NPA. And then down here has the fusion results, the level 2 ALK variants, and the RET variants right here. And so again, really good, strong concordance with both positive agreement and negative agreement, but we did have 1 discordance for each. And so this brought our PPA of 100%, but then NPA for about 97%. And so overall, when compared to the orthogonal assay, the accuracy of detection across these variant types were still really good with over 90% accuracy. The last study for accuracy was study 3, and this essentially evaluated our EGFR T790M and EGFR L858R SNVs in addition to EGFR exon 19 deletions. And again, this is just broken out. You got your level 2 EGFR, the SNVs, and the deletions. And overall, the positive concordance was really good, 23 positives to 45 expected negatives with 1 discordance. So PPA of 100% and then NPA of over 97%. So again, when compared to the orthogonal assay, the accuracy of our EGFR mutations was really good. The last study I wanted to mention was our clinical study. And so essentially this was a clinical bridging study that was conducted to essentially establish our safe and efficacy of the assay in detecting EGFR exon 20 insertions in specifically non-small cell lung cancer and identifying these patients that are good for treatment. And so the overall data set consisted of 192 patients, but the primary efficacy population was 85. These are the patients that received 200 milligrams of the drug. And you can see, and for this cohort, the overall response rate, or ORR, was about 46%, with 6 of them having a complete response and 40% of them having a partial response. Within this data set, 58 of them actually were both ODX-ET positive as well as CTA positive, giving an ORR of 41%, 5% of them having a complete response, and 36% of them having a partial response. So in summary, these results really demonstrate that the overall response rates for ODX-ET, EGFR exon 20 insertion positive population aligned with the CTA positive population, giving us really good concordance there. So in summary, I just want to hope you were able to take away and understand the breadth of testing that went into validating this assay, and just wanted to leave you a little snapshot or peek into our NGS lab and leave you with some really neat statistics. So in all, we submitted 31 formal analytical validation studies to the FDA for review. And the team extracted and processed over 12,000 DNA samples and 14,000 RNA samples. And that resulted in over 1,500 sequencing runs to provide you an FDA-approved Oncomine Dx Express Test. So thank you very much for your attention. And so now I would like to pass it over to Steve from Argent Global Services. So thank you.